Hello friends, good afternoon and welcome to EduSat live lectures. Dear friends, today in genetics we will be talking about mutations, both chromosomal and gene mutations. To discuss this topic we have with us our subject expert Dr. Charu Dogra Rawat. Dr. Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you, Amrit. Good afternoon, students. As she said, today we'll be starting with mutations. We already started with the mutations and talked about chromosomal mutations in previous sessions. We'll be continuing with the talking about mutations related to the structural aberrations which we were talking about. So the mutations, as everybody knows, is the damage in the DNA that occurs. It's a stable heritable state in the genotype. And when a large chunk of DNA is damaged or there is some kind of change that occurs in the DNA, then it is said to be the chromosomal mutations. So learning objectives for this particular session will be to understand the balanced structural mutations involving individual chromosomes or inversions, to understand about the balanced structural mutations involving multiple chromosomes or translocations, to understand the behavior of non-reciprocal and reciprocal translocations, and to understand about the fragile sites. So to start with, a chromosomal mutation that we talked about, also termed as chromosomal aberration, is a mutation involving a large segment of the genome. The structural chromosomal mutations are basically caused by the faulty repair of chromosome breaks or by the recombination between homologous but non-allelic sites. There are numerical chromosomal mutations also, which we had already talked about in our previous sessions, related to the occurrence of polyploidy when multiple sets of genome are present in the cell. And we talked about the numerical chromosomal mutations for individual chromosomes when there occurs a monosomy or a trisomy of different chromosomes which are there. So in structural chromosomal mutations, we had talked about the balanced and unbalanced structural mutations which are there. The chromosomal breaks, they occur naturally, but they can also be induced by chemical and physical agents such which are called as clastogens. So talking about balanced chromosomal mutations or unbalanced structural chromosomal mutations, they are of different in their effects. The unbalanced structural mutations involve loss or gain of genetic material and behave as partial monosomies or trisomies. That is, they show the multiple dosage effects of losing or gaining a set of contiguous genes. So when the fragment is lost, a set of contiguous genes are lost, or when the fragment is gained, then a contiguous set of genes are gained, and they therefore create an unbalanced situation in a cell. We already have talked about the two of kinds of this unbalanced structural mutations, the one being the deletion or deficiency, the structural mutations involving DNA loss, as well as we had already talked about the insertions or duplications, that is the structural mutations involving DNA gain, when a particular segment of DNA is gained in the chromosome. So there is a difference in the genome constitution of the organism, and therefore it is an unbalanced structural mutation. Today we talk about the balanced structural mutations. The balanced structural mutations are otherwise known as chromosome rearrangements, and they involve the rearrangement of DNA, but there is no net loss or gain of the genetic material. The individuals carrying such rearrangements are often phenotypically normal, but the heterozygotes may be semi-sterile because the rearranged chromosomes behave anomalously at meiosis, often segregating to yield unbalanced gametes So, the phenotypic effect of the balanced structural mutations might not be evident until and unless it is a heterozygote. So, one of the chromosome in a heterozygote might be having this kind of a mutation, and therefore, the effect will be visible. And the reason for this to occur is because the rearranged chromosomes are not segregating properly at the meiosis and resulting in the unbalanced gametes. Another reason for the occurrence of this, the effect of these mutations in the heterozygotes may be because the crossing over within the rearrangement generates unbalanced recombinant products which are non-viable. Recombination is usually suppressed at the breakpoints of such rearrangements due to steric hindrance. So, for example, a breakpoint has occurred and there is a kind of a structural rearrangement that has occurred in the chromosome. So, it will not normally recombine as it does in a normal chromosome which is there. So, the breakpoints will create steric hindrance, and recombination is usually suppressed at such breakage points. In fact, the chromosomes containing complex multiple inversions are able to suppress crossing over so that each chromosome is inherited as an intact unit. So, the entire recombination, the complete recombination is inhibited and the entire chromosome is inherited as an intact unit, and such recombination suppressors are termed as the balancer chromosomes, and they reflect the evolutionary advantages of inversions. In fact, they are also such kind of balancer chromosomes are also created in the lab to study the effect of certain mutations or the inheritance of the entire chromosomes without recombinations also. So, the balanced structural mutations usually are phenotypically normal, but the heterozygotes may be semi-sterile. We will talk about these balanced structural mutations, usually involving, the first one involving individual chromosomes, and involving individual chromosomes they are termed as inversions. Inversions are chromosome rearrangements which involve the rotation of a segment of DNA by 180 degrees within a single chromosome. So, as depicted in the picture, there is an ABCDEFG part containing chromosome, and as you can see that one part, which is the BCD part, is now inverted. So, it reads as ADCBEFG. So, the part there is no genetic loss, there is no genetic gain, but there is a rearrangement because the regions have been rearranged to give rise to an inverted chromosome. Now, if the centromere lies outside this inversion, then it is said to be paracentric inversion. That is, the inversion is occurring or the breaks is occurring only in one of the arms, as it is shown in here, that the breaks is occurring around C and E, and that portion is getting inverted. So, the arm ratio remains unchanged, the arm's length, everything remains same, but the genetic information is rearranged. So, such an inversion is termed as a paracentric inversion. If the centromere is also included in the inversion, then it is termed as pericentric inversion. So, you can see that the break is occurring around between A and B and between D and E. So, when this entire portion inverses, then the centromere is involved and the arm ratio is changed. One of the shorter arm has become a little longer and the longer arm has become a little shorter. So, this kind of inversion is termed as the pericentric inversion. Now, what are the causes of inversions that take place? The inversion occurs because of the misrepair of the chromosome breaks, because chromosome breakage is a normal phenomena. During the crossing over, when the recombination occurs, the chromosomes of the non-sister chromatids, they break and they rejoin again. But then there might occur a misrepair of the chromosome breaks that can lead to the inversion of certain segments of DNA. The intramolecular recombination between the inverted repeats. Now, in the DNA, there are repeat sequences which are present. So, the inverted repeats can recombine. When they recombine amongst themselves, then there can be an inversion that can result in. So, these are the two causes of inversions, two of the causes of inversion that can occur. Now, how do you identify these inversions? Usually, there is no phenotype, as we said that inversion results in a phenotypically normal cell unless the breakpoint interrupts a gene. So, inversion has resulted in the interruption of a gene because the breakage is occurring within a gene, and now the gene becomes non-functional, or whatever, we can assay the phenotype of the gene and it becomes different. So, we can identify an inversion because the breakpoint interrupts a gene, or the rearrangement results in unfavorable position effects, which basically means that due to the rearrangement, the particular fragment is now lying at an unfavorable position, and therefore it is exerting its effect on certain phenotype or giving a phenotypic effect, and therefore we can identify that the inversion has occurred. The inversions can be identified genotypically by their altered linkage maps. Now, genes on a particular chromosome lying at a loci might be linked, that when they are inherited, then they always tend to be together, or the crossing over does not occur between these linkage genes. But when the inversion has occurred, then two linked genes might be set apart on the chromosomes, and therefore we say that due to the altered linkage maps, genotypically one can identify that the inversion is there. In heterozygotes, for larger inversions, formation of the meiotic inversion loops is basically how the inversions are identified. Now, these inversion loops are where one chromosome twists on its axis to incorporate the reversed sequence of loci of its partner. So, when in heterozygotes there is a larger inversion which has occurred, when their two homologous chromosomes comes together, then the chromosomes of one twists on its axis to incorporate the reverse sequence of loci of the partner, resulting in the formation of this meiotic inversion loop, which is identifying that the inversion has occurred. So, the consequences of inversions. The consequences of inversions, that it's usually it does not affect meiotic segregation per se, because there is no net gain or net loss of the genetic material and the constitution of the genome remains the same. Then, as normally we can say that there is no defect on the meiotic segregation. However, it do suppress the recovery of the recombinants because of the generation of severely unbalanced crossover products. So, the segregation occurs normally, but because the products are due to inversions, there might be a deletion or duplication that has occurred. The segregation would be normal, but the products would be unbalanced, and because of this unbalanced products, the recovery of the recombinants will become difficult. The nature of the crossover products then depends upon whether the inversion spans the centromere or not. So, depending upon whether the inversion is paracentric or pericentric centromere, different kind of recombinants can be attained. For example, looking at the paracentric inversion heterozygote, if you look at this, then depends upon the number of crossovers that is occurring. So, we will see that there is a single crossover if it is happening. So, there are two chromosomes which are there. The blue chromosome is ABCDEF and the smaller chromosome is again ADCBEF. So, there is an inversion which has occurred. So, when the two homologous chromosomes will form a tetravalent, then there will be an inversion loop which will occur, and when this crossover occurs in this inversion loop, which is basically occurs in this inversion loop, then you can see that the resultant gametes. So, the effect of a single crossover within an inversion loop in a paracentric inversion heterozygote, where two altered chromosomes are produced. So, the production will be of one acentric and one dicentric chromosome. So, what do you mean by that? That if you look in the lowest panel, then there is a normal sequence that has occurred, which is a non-crossover occur. So, you know that in a tetravalent, then two of the chromatids, they do not cross over, and two of the chromatids, the crossing over takes place between the non-sister chromatids and the recombination takes place. So, when there is a single crossover between the non-sister chromatids, then two of the chromatids, there will be no crossing over that has occurred. So, they will be as normal, there will be normal sequences which will be there. And when the single crossover has occurred, then it will result into two kind of SCOs which is mentioned. One NCO's SCO will be dicentric, that it will have two centromeres which is there. So, there might be a duplication and deletion that has occurred, and there will be an SCO where it will be an acentric, that it will not contain any centromere which is there, but there is also again duplication and deletion of the sequences that has occurred. Acentric chromosome is usually lost because it has no means to attach to the spindle, no centromere, no attachment to the spindle. So, acentric chromosome is usually lost. Dicentric fragment is stretched between the two nuclei and is either excluded due to the anaphase lag. So, when the anaphase condition occurs, that because of the difference in the stretching of the chromosome towards the two poles containing the two centromeres together, there occurs an anaphase lag, or this dicentric chromosome can be fragmented randomly, generating a broken end that may imitate a broken fusion bridge cycle. So, we will look at this broken fusion bridge cycle in a short while, but basically what it is trying to say is that the recovery of recombinants is a little difficult. So, when during the paracentric inversion has occurred, then there is an occurrence of a dicentric and an acentric chromosome or chromatids. The dicentric chromatids either will be fragmented or will be lost or excluded due to the anaphase lag, and the acentric chromosome will in any case be lost because it will not be able to go to any of the poles because there will be no place for the spindle to be attached. If we look at the pericentric inversion heterozygote, then what happens? The effect of the crossover in a pericentric inversion heterozygote where two altered chromosomes are produced, both of which contains duplicated and deficient regions. So, you can see that when the tetravalent thing has been formed, then the inversion loop including the crossover, then there are four chromatids which are there. So, there will be two normal sequences, there will be one normal sequence and one inverted sequence which was coming from the parent. There is a single crossover which will contain a duplication and a deletion, and another single crossover which will again contain some duplication and deletion, but all of them will be monocentric in chromosomes which are there. So, the nature of the recombinants that occur after a paracentric or a pericentric inversion heterozygote are different, but the recovery of the recombinants is difficult in both the cases. If crossing over always occurred within a paracentric or pericentric inversion, the 50% of the gametes would be ineffective. The viability of the resulting zygotes is therefore greatly diminished. So, the structural mutation is said to be balanced because there is no loss or no gain of the genome or the genetic content which is there. But inversions might lead to the production of the unbalanced gametes, and the production of these unbalanced gametes will affect the viability of the resulting zygotes, which is greatly diminished. Furthermore, up to 1/2 of the viable gametes have the inverted chromosome, and the inversion will be perpetuated within the species. The cycle will be repeated continuously during meiosis in future generations. So, whether it is a paracentric or pericentric inversion that has occurred, there is a difficulty in the recovery of the recombinants. There is a diminished viability of the resulting zygote when such unbalanced gametes are used, and there is a perpetuation of the inversion in the species that occurs and therefore passed on to the future generations also. There is a special kind of chromosome when we are talking about the intramolecular structural mutations, then these are termed as ring chromosomes. They occur due to intramolecular chromosomal aberration when a chromosome which has lost telomeres from both the ends fuses the broken ends together. So, actually ring chromosomes can occur in two manners. The one thing as shown here is that the telomeres break apart, and then the two ends fuses together to give rise to what is called as the ring chromosome. The acentric segment is usually lost and the ring chromosome is what remains. The other way is that probably the ends of two different chromosomes can also be lost, and then they can be fused together to give rise to the ring chromosomes also. But that is a very rarer situation, and this is the most common situation in which the telomeres of a single chromosome of an individual chromosome is lost and it fuses together to give rise to the ring chromosomes. Now, ring chromosomes when replicate, then a ring chromosome at cell division, if a ring replicates itself with no sister, so there are three possibilities that can occur. If a ring replicates itself with no sister chromatid exchange in prophase, the ring chromatids separate at anaphase, resulting in equal sized rings as shown in the topmost panel. So, the entire ring chromosome has replicated and then they are separated into the two chromosomes which are there. Another situation is there if one crossover event has occurred, the two daughter chromatids form a dicentric bubble sized ring as the centromere gets pulled to opposite poles, and depending on whether breakage occurs, deletions and duplications result. So, in this middle panel it is shown that a single crossover has occurred and the two daughter chromatids had formed a dicentric double sized ring. So, actually the two rings have fused together sort of a things, and there are two centromeres, and these two centromeres then during the anaphase gets pulled towards the two poles, and then there can be a breakage and fusion, and again that breakage fusion cycle that can resume and resulting in the deletions and duplications which is there. If there are two sister chromatids, again there is a double crossover, it would result in interlocked rings called as the concatenations with subsequent asymmetric breakage leading again to the unbalanced products. So, this ring chromosomes does occur in autosomes. There are ring chromosome 18, there are ring chromosome, there are many genetic disorders in human which is because of the ring chromosomes, and this ring chromosomes can occur in the cell cycle. They can behave in these three manners leading to again different kind of chromosomal aberration which is there. Now, coming to the balanced structural mutations that involve multiple chromosomes. Translocations are structural aberrations in which part of one chromosome is transferred to another. There are two types of translocation. A non-reciprocal translocation, it involves the one-way transfer of the material, that is from one non-homologous chromosome, the part is transferred to another non-homologous chromosome, and this is termed as the non-reciprocal translocation. In the reciprocal translocation, as depicted on the right hand panel of the picture, it occurs when two chromosomes are broken and the digital fragments are swapped and rejoined. So, basically there is a swapping and rejoining. So, there is a rearrangement in both the chromosomes that has occurred. So, that is termed as a reciprocal translocation. There is another term which is used termed as internal translocation. This occurs when a chromosome segment is inserted into an interstitial site within another chromosome. And a terminal translocation, it involves material added onto the end of the another chromosome. There is another term which is used called as the jumping translocation, in which one of the fragment jumps in over multiple chromosomes and then shows the kind of effect which is there. So, it jumps to one of the chromosome, then to the other chromosome. It can be interstitial or terminal translocation, but it has jumped to multiple chromosomes, then it is said to be a jumping translocation which is there. Now, looking at the non-reciprocal translocation, there is a special term which is used called as a Robertsonian translocation or whole arm translocation which is there. It is an extreme form of non-reciprocal translocation where two acrocentric chromosomes are fused at or near the centromere to generate a compound chromosome which segregates as a single unit. So, you can see that there are two normal non-homologous acrocentric chromosomes where the centromere is present towards one end. So, one of the arm is shorter and one arm is longer, that is termed as the acrocentric chromosomes. When two non-homologous acrocentric chromosomes are involved and there is a non-reciprocal translocation, then one of the chromosomes is said to be a compound chromosome because now it contains one centromere and two arms of both the chromosomes. Ideally the entire two chromosomes are there and a little smaller fragment is usually lost which does not give rise to any kind of a phenotypic effect. So compound chromosome, I would just like to emphasize here, is the two whole chromosome fused together. The loss of part of the short arm appears to have no phenotypic effect. A submetacentric chromosome is generated containing the long arms of both original acrocentric chromosomes and a common centromere. So you can see that that is termed as a compound chromosome. Here I would like to mention about another chromosome which is said to be an attached chromosomes. We will talk about this more later, but attached chromosomes are when two submetacentric chromosomes such as X chromosomes join together. Such chromosomes are dicentric. So the difference between a compound chromosome and a attached chromosome is that a compound chromosome contains the entire two chromosomes but with one centromere which is there. A submetacentric chromosome is there and usually occurring due to the because of the Robertsonian translocation, whereas attached chromosomes are when two chromosomes, submetachromosomes join together, actually attached to each other, containing both the dicentric or both the centromeres which are present there are present. So the Robertsonian translocation in humans is found in a familial Down syndrome. So we know already about the Down syndrome, which is trisomy 21. So in this case, basically the chromosome number 21 is in trisomic condition because of the translocation that has occurred and not due to the normal non-disjunction which is there. So most of the 95% of the Down syndrome is because of the non-disjunction of the chromosome number 21. So that one of the gametes contain two chromosomes and one of the gamete contain one chromosome, and after fusion with the other gamete, they give rise to a trisomic condition, and that is termed as the Down syndrome which is there. In this case, we will see that there will be trisomy 21 occurring due to the translocation and not due to the disjunction, and it is said to be familial because this kind of Down syndrome is inherited from generation to generation. The normal Down syndrome, which is occurring due to the non-disjunction of chromosomes, is usually not passed on from generation to generation, but this is found in the families and runs from the generation to generation. So the recurrence risk of Down syndrome is very low when caused by the spontaneous non-disjunction, but much higher when caused by a Robertsonian translocation involving chromosome number 21. The familial Down syndrome is caused when one of the parents contain a 14-21 DG translocation. So this is a Robertsonian translocation. The 14 chromosome belong to the group D and 21 chromosome belongs to the group G. So when a fragment of 14 chromosome is attached to the 21 chromosome, then there occurs a familial Down syndrome which is there. So you can see in normal condition there are two chromosomes, the 21 chromosome and the 14 chromosome which is there, and therefore they give rise to the normal gametes in which a single copy of both the gametes are present. When the translocation carrier is there, then it contains a part, basically the part of the 21 chromosome is attached to the chromosome number 14. So you can see the translocation carrier has a single 21 chromosome, however has a double because of the translocation of one of the 21 chromosome onto the chromosome number 14 which is found. So when the gametes are formed, these kind of gametes occur, and when they fuse with a normal gamete, then they give rise to normal individuals which will contain, the 46 number of chromosomes which is there. They will also give rise to the translocation carrier, which is again, when the translocated chromosome combines with the normal 21 chromosome which is there. So it contains the complete genome. There are two parts of the 21 chromosome and two 14 chromosomes, although one of the 21 chromosome fragment is translocated onto the 14 chromosome, but the genetic content is constant and therefore it is a translocation carrier. But in other conditions there occurs a trisomy 21, where there are two 21 chromosomes which are present and then one part of the 21 chromosome which is also present into the 14 chromosome which is there. So this is a kind of a Down syndrome which is exhibiting trisomy 21, but this is a different kind called as the familial Down syndrome. And in the monosomic condition, which is usually lethal, where there is only one copy of the 21 chromosome is present. The individual is phenotypically normal even though he or she has only 5 chromosomes and is referred to as a balanced translocation carrier. So, they have 45 chromosomes, but the genetic content is normal because 2 sets of genes present on the 21 chromosomes are present as well as the 14 chromosome are present. So, that is said to be a balanced translocation carrier. During meiosis, one of the gametes contain 2 copies of the chromosomes, a normal chromosome and a second copy translocated to the chromosome number 14. Once such a gamete is fertilized by a standard haploid gamete, the resulting zygote has 46 chromosomes but 3 copies of chromosome number 21, and these individuals exhibit Down syndrome as shown. So, this was all about the non-reciprocal translocation. The behavior of the reciprocal translocation, if we see at the meiosis, then these are the possible origin of reciprocal translocation that can occur when the 2 segments are mutually exchanged between 2 non-homologous chromosomes. They at synapses, they result in the formation of this tetravalent which is there, and there are 2 possible segregation patterns leading to the gamete formation. One is termed as the alternate segregation, where the normal chromosomes, the parental chromosomes, and basically the balanced translocation chromosomes are segregated. This is termed as the alternate segregation. There is another segregation that can occur which is termed as the adjacent one segregation, in which one of the normal chromosomes segregates with one of the copy of the translocated chromosome. So, the genetic content will be duplicated and deficient, and in the other again the number 1 and number 3 have translocated as shown in the lower panel. That is again the content is duplicated and some of the deficiency is there. There is another kind of segregation that can occur, adjacent 2 segregation, which is very rare, in which basically 2 homologous chromosomes goes to the one of the poles which is there. So, this is about the reciprocal translocation which is there. The last would be the fragile sites which we talk about. The unstained chromosome gaps identified as secondary constriction induced by stressful growth conditions are thought to be the hotspots of chromosome breakage and are termed fragile sites. In humans, over 50 common fragile sites have been identified which are present in most people. About 20 fragile sites described which are present in minority of the individuals. Generally, these fragile sites have no phenotypic effect. However, one of the phenotypic effect is shown in the fragile X syndrome, also called as the Martin-Bell syndrome. You can see with the arrow the non-stained part which is there. The folate sensitive rare fragile site at XQ27 that corresponds to the presence of mental retardation, large testes and characteristic facial features in males, and mental retardation in some female carriers which is there. The cause of the syndrome is a polymorphic triplet repeat mutation in the gene termed as FMR1. In FMR1, the trinucleotide sequence CGG is repeated in an untranslated area adjacent to the coding sequence of the gene called the upstream region. The number of the repeats varies within the human population, and a higher number correlates directly with the expression of the fragile X syndrome. Normal individuals have between 6 to 54 repeats, whereas those with 55 to 230 are the carriers. They are unaffected, but they are considered carriers. More than 230 repeats leads to the expression of the syndrome. So, these are the sites which are basically susceptible to the chromosomal breakage which are there. Thank you. In this session, we will talk about gene mutations, the damage to the DNA sequence that occurs within a gene. So, learning objectives for this session would be to understand more about the gene mutations, to understand about the spontaneous and induced mutations, and to understand various types of mutations based on location of the mutation, the type of change that they confer, and the phenotypic effects that they produced. We will just talk about the overview or the terminology of the gene mutations which is there, and deeper we will go into the examples and types in our later sessions. So, a gene mutation is a stable, heritable change in genotype caused by an alteration to the nucleotide sequence within a gene of the genome. A gene, genome, cell or individual carrying such a given mutation is termed as a mutant. The gene mutations can affect the nature of the gene product, or it can also interfere with its gene expression. The gene mutations convert one allelic form of a gene into another. For many gene loci, there is a wild type allele which predominates in the population because it confers the greatest fitness, which is basically the ability to survive and reproduce. This generally encodes the normal functional product associated with the gene, and the wild type phenotype reflects the normal gene activity. So, normal gene in a cell having normal gene activity is termed as the wild type allele and is said to exhibit the wild type phenotype. Other rare alleles are designated mutant alleles, and the quantity and or structural properties of the encoded product may differ, generating distinct mutant phenotypes which are there. So, not normal alleles are the mutant alleles, and usually either the quantity differs or their structural properties differ of the encoded product differs, and they are said to exhibit the mutant phenotype which is there. The gene mutations away from the wild type are usually deleterious or selectively neutral, however few are beneficial also. So, we will talk about all these different types of gene mutations which are there. So, to start with, there are different ways to classify these gene mutations. We will talk about the spontaneous mutations first. Spontaneous mutations are generated by lesions or the damaged DNA that arise due to intrinsic properties of the DNA molecule or interaction with DNA damaging chemical agents such as free radical produced as a byproduct of the metabolism. So, spontaneous mutations is basically when you do not know any reason. It is basically due to the intrinsic property of a DNA that the damage in the DNA has occurred. There might be an error in the recombination process, there might be an error in the repair process, but intrinsically this damage to the DNA has occurred. In fact, the mutagens which are which can act as mutagens can be metabolic products which can be there inside the cell. So, that can be called as a induced mutation, but we talk of it as a spontaneous mutations because the mutagen is actually produced by the cell itself. It is not artificially added. So, when we add artificial mutagen, it is said to be an induced mutation, otherwise it is a spontaneous mutation. So, basically the distinction becomes a little blur, but that is how we deal with it. So, spontaneous mutations can also arise, as I said, through recombination and replication errors and the activities of what are termed as the mobile genetic elements. So, there are various transposons which are present which can jump from one position to the another, and they can also cause the damage to the genes which are there, and therefore they can cause spontaneous mutations which are there. The spontaneous mutation rate is exceedingly low for all the organisms. The rate varies between different organisms and even within the same species. The spontaneous mutation rate varies from gene to gene. So, what is the mutation rate? The mutation rate is defined as the likelihood that a gene will undergo a mutation in a single generation or in forming a single gamete. So, this mutation rate, the spontaneous occurring of the mutation in a particular generation is usually very low in the organisms, however differs within the species and outside the species and within the species from gene to gene also. The variation between genes in a given organism may be due to the inherent differences in the mutability in different regions of the genome. So, some DNA sequences appear to be highly susceptible to mutations and known as mutation hotspots. So, the spontaneous mutations, as we said, can vary from gene to gene, and this can be due to the intrinsic property of the DNA encompassed in those genes. So, there are certain DNA sequences which are more susceptible to mutations and are termed as mutations hotspots. The variability between different organisms may in part reflect the relative efficiencies of their DNA proofreading and repair systems which are there. So, the spontaneous mutations, if you look at this thing, then they differ, the mutation rate differs in different organisms. The viral and bacterial genes undergo spontaneous mutation at an average about 1 in 100 million replications or cell divisions. Maize, Drosophila and humans demonstrate rates several orders of magnitude higher. The genes studied in these groups average between 1 in 10 raised to the power of minus 6 and 1 in 10 raised to the power of minus 5 mutations per gamete formed. Some mouse genes are another order of magnitude higher in their spontaneous mutation rate, where they show 1 in 10,000 or 1 in 100,000 replications or cell divisions, the spontaneous mutations are found. So, there is a variability in the mutation rate in different species and within one species from gene to gene that occur. So, we talk about the induced mutations. They are generated by lesions that are caused by the application of DNA damaging agents termed as mutagens. So, usually induced mutations we talk about when the DNA damaging agents are external. They can be natural or artificial, but that is an external application of the DNA damaging agent. When the DNA damaging agents are produced by the cell and they induce mutations in the DNA, then that falls again in the part of the spontaneous mutations only. The mutagens can be chemical, physical or biological agents. We look at some of the common mutagens. The chemical mutagens can be deaminating agents, for example, nitrous acid, which is non-specific, and sodium bisulfite, which specifically deaminates cytosine. So, we will talk about the mechanisms of mutation, or in fact that has been covered in the molecular biology series also, that how the mutations are caused, and one of the mechanism is by the deamination which is there. So, nitrous acid and sodium bisulfite causes are the deaminating agents that causes mutations. Alkylating agents such as ethyl methylene sulfonate EMS and dimethyl nitrosamine, they are alkylating agents which are there. Then the donors of bulky addition products, which is aflatoxin, B1 and benzopyrene, when metabolized, they form reactive species which add bulky chemical groups to the bases, and this addition of the bulky groups again causes mutations that can lead to the mutations which are there. The base analogues such as bromodeoxyuracil which is there, the intercalating agents such as ethidium bromide and acridine orange, which can intercalate between the two strands or between the bases and therefore disrupt the base pairing or cause the mutations. The cross-linking agents such as the bifunctional alkylating agents, the nitrogen and sulfur mustards and platinum derivatives which are there. The planar molecules or the SORA lanes which are called as are also act as the cross-linking agents, and they are the chemical mutagens. Looking at the physical mutagens, the ionizing radiations, they cause a variety of DNA lesions including damaged bases, damaged sugar rings, nicks and breaks. The effect can be direct, that directly they act on the DNA, or they can also they can also serve to generate the reactive oxygen species, which then can act as the chemical mutagen for the mutagenesis to be there. UV radiations can also induce lesions. The UV induced lesions are termed as photoproducts. The predominant effect generation is of the generation of photodimers such as cyclobutyl pyrimidine dimers, and there are another set of dimers which are also there. The biological mutagens are restriction enzymes. They induce breaks into the invading genomes, for example, a virus infecting a bacteria, and the bacteria has this restriction enzymes which will invade its genomes and induce breaks and causes mutations so that the virus is not able to replicate inside the bacteria. The mobile genetic elements such as the viruses, the bacteriophage mu and retroviruses, the episomal plasmids such as F factor, and transposable genetic elements such as P elements or TY elements, which are basically jumping from one fragment to the another, causing the mutations to occur. So basically the mutagens compasses chemical, physical and biological mutagens which are there. Now looking at or finding out that are these mutations random or adaptive. So looking at the spontaneous mutation rate, one thing to identify was whether these mutations are basically arising, they are spontaneous, they are occurring randomly, or whether due to the presence of certain chemical substances or presence of certain conditions, the mutations are being induced. So there was a fluctuation test which was devised by Luria Delbrück, and which tells us whether the mutations occur randomly, spontaneously and randomly, or they are induced or present or occur more in certain conditions which is there. So what they did was they took E. coli and they grow it in a bulk flask, and then they also grow it into the individual colonies. So E. coli is grown in the absence of the T1 phages. When each of the samples becomes 20 million cells, so then each sample they take 20 million cells of this E. coli and infect in the petri dishes containing the T1 bacteriophages, and then they are allowed to grow. So when the bulk culture, the samples are taken and tend to be grown, then there is usually a relatively even distribution of colonies which is there, a resistant colonies towards the T1 bacteriophage. So if you have a bacteriophage on a petri plate and the E. coli is grown and put on the plate, the E. coli will be able to survive only if it is resistant to that bacteriophage. If the bacteriophage infects the bacteria, it will lyse the cell and there will be no growth. So in the petri plates, if you can see on the left hand panel, the 20 million cells in all the different 10 samples which were taken, more or less there is an equal distribution of colonies on each of the plate which is there. However, when the samples were taken as there are individual cultures which are taken, and then these are plated, then there is a great fluctuation in the number of ton resistant colonies or bacteriophage resistant colonies which are there. So some of the planes on the right hand panel, when the 20 petri plates are shown, then some of the plates does not have any E. coli, and some of the plates there are accumulation of the E. coli cells which is there. So this is a normal fluctuation test. Now the hypothesis is basically if the mutations are said to be adaptive, then because the number of cells which are taken and the number of bacteriophage which is plated is constant in each of the plates, if their mutations were supposed to be induced, then there will be a equal distribution of colonies that will become resistant. There will not be any bias, there will not be any fluctuation, because there are same number of E. coli which is taken and same number of bacteriophage which is taken. However, if the mutations are random, if the mutations are occurring randomly, then the number of colonies that will appear in plates will differ. The number of colony in which the mutation has occurred earlier will produce much more progeny, and therefore there will be much more colonies in those plates. And in some of the plates where the mutation has occurred lately, then there will be less number of colonies in the plate which will be there. And some of the plates even does not have any colony which is resistant to the bacteriophage. So this fluctuation in the recovery of the resistant E. coli tends to state that the mutation is random and it is not adaptive. So the other figure also explains the same thing more clearly, that if the mutation is induced by the bacteriophage which is present in the petri plate, then more or less in all the plates there will be a constant number of resistant colonies that will appear, because the same number of E. coli has been added and same number of bacteriophage that has been plated. However, if the mutation was spontaneous, then in the ones where the mutation has occurred earlier, more progeny will be produced, and therefore the petri plate will contain lot of resistant colonies. But in some of the colonies where the mutations occurred later, there will be less number of colonies, and in some plates there will be no resistant colonies at all, because the mutation was random and it did not occur in those particular colonies which is there. So, this is a very good fluctuation test which tells us that the mutations are random. However, there still goes on a debate about this, but this test definitely clarifies this point. Now, we will talk about the terminology of mutations based upon the location or the nature of change they produce or the phenotypic effects which they are producing. So, mutations based on the location, the somatic mutations are those occurring in any cell in the body except the germ cells. The mutations arising in the somatic cells are not transmitted to the future generations. When a recessive autosomal mutation, now autosomal mutation or sex-linked mutations, autosomal mutations are the mutations which are occurring on the chromosomes which are autosomes, which are not X and Y, and X-linked mutation or Y-linked mutation when a change or damage in the DNA occurring in the X or Y chromosome which is there. So, when the autosomal mutation occurs, when a recessive autosomal mutation occurs in a somatic cell of a diploid organism, it is unlikely to result in a detectable phenotype. The expression of most such mutations is likely to be masked by the expression of the wild type allele within that cell. The somatic mutations will have a greater impact if they are dominant, or in males if they are X-linked, since such mutations are most likely to be immediately expressed. The impact of dominant X-linked somatic mutations will be more noticeable if they occur early in the development, when a small number of undifferentiated cells replicate to give rise to several differentiated tissues or organs which are there. The germinal or germline mutations are those occurring in the germline or gamete forming tissue. They may be transmitted to the offspring as gametes, and they have the potential of being exposed in all cells of the organisms. Inherited dominant autosomal mutations will be expressed phenotypically in the first generation. X-linked recessive mutations arising in the gametes of a homogametic female may be expressed in hemizygous male offspring. This will occur provided that the male offspring receives the affected X chromosome. The occurrence of an autosomal recessive mutation in the gametes of either males or females may go unnoticed for many generations until the resultant allele has become widespread in the population. Usually, the new allele will become evident only when a chance mating brings two copies of together into the homozygous condition. All these detection of mutations will be more clearer after the session of pedigree analysis is conducted, because pedigree analysis we deal with such mutations and we look at the families that where the mutation is occurring. The mutations based on the type of change, there are point mutations or base mutations, a change of one base pair to another of a DNA molecule. If a pyrimidine replaces a pyrimidine or a purine replaces a purine, it is termed as transition. If a purine replaces a pyrimidine, it is termed as transversion. A change of one nucleotide of a triplet within a protein coding portion of a gene may result in the creation of a new triplet that codes for a different amino acid in the protein product. If this occurs, the mutation is known as a missense mutation. One gene product transformed into another gene product. It is termed as missense mutation. A second possible outcome is that the triplet will be changed into a stop codon, the termination of translation of the protein. It is termed as a nonsense mutation. If the point mutation alters a codon but does not result in the change in the amino acid product because of the degeneracy of the genetic code, it can be considered as a silent mutation which is there. Then there are frame shift mutations. They involve the insertion or deletion of one or more nucleotides at any point within the gene. The loss or addition of a single nucleotide causes all of the subsequent 3-letter codons to be changed. Like you can see, the cat saw the dog. If there is a change of one letter, there is a substitution. So, there can be the bat saw the dog, the cat saw the hog, the cat sat the dog. If the cat becomes bat, then there can be an alteration in the gene product, and these are the cases of point mutation. There can be a loss of one letter. If the C is lost, then the entire reading frame is changed, and that is said to be the frame shift mutation. Or if there is a gain of one letter, for example, the insertion of M, then again the reading frame is changed. You cannot read again the cat saw the dog. Therefore, that is termed as a frame shift mutation. The frame of the triplet reading frame is altered. Then there are certain terms used for the mutation based on the phenotypic effects. A loss of function mutation is one that reduces or eliminates the function of the gene product. Mutations that result in complete loss of function are known as the null mutations. In diploid organisms, haploinsufficiency is a condition that occurs when the single functional copy of the gene does not produce enough gene product to bring about a wild type phenotype. So, that is termed as the haploinsufficiency, and in humans, Marfan syndrome is an example of a disorder caused by haploinsufficiency. In this case, as a result of loss of function mutation in one copy of the FBN1 gene which is there. Then a gain of function mutation results in a gene product with enhanced or new functions. Most gain of function mutations are dominant. Then there are certain mutations which are termed as visible mutations. They are recognized by their ability to alter a normal or visible wild type phenotype. For example, all of Mendel's P characteristics and many genetic variations encountered in Drosophila fit that designation, since they cause obvious changes to the morphology of the organism which is there. Some mutations give rise to nutritional or biochemical effects. For example, in bacteria and fungi, a typical nutrition mutation results in the loss of ability to synthesize an amino acid or vitamin. In humans, sickle cell anemia and hemophilia are examples of diseases resulting from biochemical mutations. Still, another category consists of mutation that affects the behavioral pattern of organisms. The primary effect of behavioral mutations is often difficult to analyze. For example, the mating behavior of a fruit fly may be impaired if it cannot beat its wings. However, the defect may be in the flight muscles, nerves leading to them, or the brain, where the nerve impulses that initiate wing movements originate. The regulatory mutations, they affect the regulation of the gene expression. A mutation in a regulatory gene or a gene control region can disrupt normal regulatory processes and inappropriately activate or inactivate expression of a gene. So, these are the different terminologies which are used to refer gene mutations. We will take up the examples and go deeper into the mechanism of how these gene mutations occur in our subsequent sessions.
On that note, I would like to thank ma'am for this very interesting discussion, and I would like to thank you, dear friends, for watching our show. I hope you have come to understand what gene mutations and what chromosomal mutations are all about, and you have come to understand its various types and its various examples. On that note, thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.